So what we're going to present here is not an official opinion of Michigan State University or the University of New England. I'm Zach Burton, and my co-host is Lei Lei, and we're completely responsible for content. So this is a Life on Earth, Origin of Life presentation. And today we're going to discuss type 2 transfer RNA evolution, which has been solved as a puzzle. And it's not that hard of a puzzle, particularly if you have the answer in front of you. Okay, Lei Lei, School of Biological Sciences, University of New England. And I'm Zach Burton, Michigan State University, now retired, and I was in the Department of Biochemistry and Molecular Biology. So these are the subliminal messages we want to impart. So it's a story about transfer RNA evolution. It's a story about evolutionary intelligence. So what do we mean by that? So evolution learns to solve new problems and to take on new capacities. So it's very much like computer learning, artificial intelligence or machine learning, if you want to think about it that way, language, logic, linguistics. This is RNA linguistics with A, G, C, and U as the relevant letters. What we're showing is, I think, the greatest story in 4 billion years. So as long as there's been some pre-biology or biology on life. And it's a puzzle just about anybody can solve. So you don't need to be a biochemist. Some people don't like evolution and they don't like biology, but they like language and intelligence and linguistics and logic and things like that. So this can be for you too. So to origin of life problems, there are bottom-up and top-down presentations. And bottom-up approach is to make life in a test tube, for instance, which is still a challenge, but maybe one that's becoming an easier challenge with time. And a top-down approach is to infer origin of life from existing biology, in this case, tRNA sequences. So we'll present the 331 nucleotide mini-helix theorem. It's a theorem and not a model because it's a proven hypothesis. So you can be absolutely certain that this is true and correct, and you can use this as a cheat sheet to solve the puzzle. So this is recorded history from about 4 billion years ago, and the history was recorded in genetic code and it was never overwritten. So what we're doing here is we're identifying root sequences of transfer RNA, type 1 and type 2 transfer RNA. And I'm going to say that transfer RNA is a collectible, practically an antique at an age of 4 billion years. So we can address questions similar to this. So is there a God? So that's not really a good scientific question, and we don't have much to offer on that. Is a God necessary? So it appears that gods are not necessary to evolve tRNA, that you can generate tRNA by a straightforward chemical evolution process. And we provide many details about that process. So most transfer RNAs are type 1 and some are type 2. And we'll show you how you count the type 2 tRNAs in archaea and bacteria. Type 2 tRNA have a longer variable loop. So the variable loop is initially 14 nucleotides, which is 7 plus 7, which is actually relevant as it turns out, instead of 5. So in a type 1 tRNA, the variable loop is 5 nucleotides initially. In a type 2 tRNA, it's 14. If you have 14 nucleotides, which is 7 plus 7, and you delete 9, you're left with 5. That's how this is generated. This was generated out of this sequence here. So it turns out there are 3 set points for a type 2 variable loop. And you can name these 0, 1, and 2, show you what that means in a minute. In archaea, transfer RNA for leucine has a score of 2, and transfer RNA for serine has a score of 1. So as it comes out, this number cannot equal that number. This is universal for archaea. The score is always 2 for tRNA leucine and always 1 for tRNA serine. And in bacteria, there are 3 type 2 tRNAs, not 2. And tRNA tyrosine has taken over this 2 position. So it gets a score of 2. tRNA leucine is downgraded from a score of 2 to a score of 1. And tRNA serine is downgraded from a score of 1 to a score of 0. Once again, this number does not equal that number, does not equal that number. And there's a reason for that. So the story we're imparting is one about life, how life evolved on planet Earth. Transfer RNA, we're going to describe its chemical evolution. Transfer RNA is the genetic adapter, and you need a genetic adapter. Otherwise, you don't have a genetic code, and without a genetic code, you can't have biological complexity or diversity. And we have a system that will generate polyglycine in an RNA-dependent reaction. And once you have that kind of a reaction, you can evolve to a protein synthesis system that can synthesize a myriad of proteins and give you biological diversity and complexity. So root sequences of transfer RNA, primordial transfer RNA type 1 and type 2, so these are pre-life sequences, are known with essential certainty, very few ambiguities. The sequences are completely ordered, that is 100% RNA repeats and inverted repeats, as I'll show you. And type 1 and type 2 primordial tRNA sequences embedded in their sequences is a rather compelling story about the origin of life. So we're going to give lots and lots of detail about the origin of life on this planet. Transfer RNA is the genetic adapter. And what that means is it links nucleotide sequence to amino acid sequence, which is protein sequence, at a ratio of 3 nucleotides to 1 amino acid. And that's sort of the coding basis for all life on this planet. Transfer RNA, we're going to refer to, at least briefly and metaphorically, as the God molecule. And the transfer RNA anticodon loop is the God loop within the God molecule. So God confers life on Earth. Transfer RNA appears to fulfill that function, and the transfer RNA would not be able to fulfill this function if not for the tRNA anticodon loop, which is a highly specialized loop to present a 3 nucleotide anticodon to encode a single amino acid. So we'll show the 331 nucleotide mini-helix theorem, and this is a theorem, not a model, because it's a proven hypothesis. And you can rely on this theorem completely. And when you look at this sequence, you're looking at the cheat sheet to solve the puzzle that we're presenting today. But you're also looking at a story for how life evolved on Earth 4 billion years ago. Type 2 transfer RNA. So first, let's look at type 1 tRNA, which we talked about in a previous presentation. This is type 1 tRNA because it has a short variable loop. It's 5 nucleotides. So 1, 2, 3, 4, 5 nucleotides. And this sequence has to be expanded in a type 2 tRNA, and it's expanded like this. So you see this chicken wing coming off the transfer RNA molecule. This is 7, and that's 7. 7 plus 7 is 14. These 7 relate in sequence to these 7, and these 7 here relate to these 7 here. In terms of this is a tRNA leucine from an ancient organism. This organism is very close to being the last universal common ancestor on Earth. So it's an ancient living fossil, if you want to think about it that way. So these 7 bases here, these are related to a 3' acceptor stem. This is based on a CGC repeat, complementary GCG repeat. These 5 here correspond to those 5 there, and these 5 here. And this is based on a GCG repeat. And the D loop is based on a UAGCC repeat, and then you have a stem loop stem, stems of 5, loop of 7, stem loop stem, stems of 5, loop of 7, U-turn after a U-turn after a U. So this sequence here and this sequence here are homologs. That means they're close to being identical twins. And here you have ACCA, 2 C's to hold, in this case, tRNA leucine with a leucine attached at the ribose ring next to a peptide chain attached to another tRNA, and you can transfer leucine to the growing protein chain to build a protein. A is the primitive discriminator base. So initially ACCA was an adapter to attach amino acids to make primitive proteins. So these are cloverleaf diagrams, and you sort of have to get used to looking at them. It's like a flattened version of this image, if you want to think about it that way. In a previous webcast, we described all this part of the molecule. So here we'll just try to describe this part and that part. And we're in archaea now. So this is transfer RNA leucine, and this is transfer RNA serine. LURS is the enzyme that puts leucine here, does not put leucine there. CRRS puts serine here, does not put serine here. Try to explain a little bit about how that works. I promised you that a tRNA leucine had a score of 2. That relates to these 2 bases, which are unpaired between the 3' variable stem. The V arm is a stem loop stem, and then a number of unpaired bases. This is called the Levitt base pair. It's a reverse Watson-Crick base pair. That means it's like a Watson-Crick base pair in DNA, but with one of the bases flipped over. So this is a score of 2. This is a score of 1. That's true in Pyrococcus. It's true in all archaea. And this score cannot equal that score for an archaea or a bacteria. In this case, LURS interacts with this A and this G on the loop, and that's part of the determinant that it uses to place leucine accurately here. And LURS is not attracted to these 2 U's. So when you see T, that's a DNA sequence. So in RNA, that's a U. So UU is not attractive to LURS. But LURS can't reach these 2 U's anyway because of the trajectory of this arm coming off the body of the tRNA. On the other hand, CRRS is not attracted to this arm. CRRS lies across this arm and interacts with the T-loop. And so CRRS makes its own set of contacts to put serine here. CRRS can't interact with this arm because this arm is positioned too far underneath the T loop. Can't reach. Oh, I should show you this. So UAGCC repeat, the D loop sequence is based on a UAGCC repeat. So here you have UAGCC, UAGCC, two perfect repeats, U, G for A, GCC, UA. That's almost a perfect UAGCC repeat, just like in the model, but then the model is correct. So this is bacteria now, and so we're going to look at Thermus thermophilus. Thermus is an ancient bacterium, and it's a good reference species for the oldest bacteria. So here you have the Leavitt base pair, and here you have one unpaired base. And here you have the Leavitt base pair and 0 unpaired bases. So this is tRNA for leucine, to place leucine. This is tRNA for serine. And serine has a score of 0 in bacteria. Leucine has a score of 1. So it's been downgraded from its score in archaea by 1. And this one has been downgraded to 0 from 1. 1 in archaea, 0 in Thermus. And this is in bacteria. So tRNA tyrosine gets a score of 2. So it bumped leucine tRNA out of the 2 position in evolution. It's got 2 unpaired bases between this stem and the Leavitt base pair there. And tyrosine aminoacyl tRNA synthetase, the enzyme that puts tyrosine there, interacts with this G and that U on the loop. And it can reach them because of the attitude of the loop. With a score of 2, puts it in reach to contact those 2 bases. So there are 3 set points for type 2 variable loops in bacteria and archaea with scores of 0, 1, and 2. In archaea, tRNA leucine gets a score of 2, tRNA serine a score of 1. In bacteria, tRNA tyrosine gets a score of 2, tRNA leucine is downgraded from 2 to 1, and tRNA serine is downgraded from a score of 1 to 0. This number cannot equal that number, cannot equal that number cannot equal that number. Otherwise, there's confusion in aminoacylation of tRNAs. So in Pyrococcus, you have this as the primordial type 2 tRNA sequence, and you have tRNA leucine and tRNA serine. And in the primordial tRNA, you have a 3' acceptor stem. That's a 3' acceptor stem ligated to a 5' acceptor stem, like that. So CGC repeat ligated to a GCG repeat. And in tRNA leucine, you have a few sequence changes, but again, you have 7 plus 7, and this forms a stem loop stem with 2 bases to interact with LURS, the charging enzyme for this tRNA. And tRNA serine, you have a 1 base insert, so this sequence is 15 instead of 14. And it's got a stem loop stem, but it's got a score of 1. This one has a score of 2. And so that's how they're discriminated for accurate tRNA charging. Basically, this sequence is very close to that sequence, is very close to that sequence. Pretty clearly, this sequence and this sequence are derived from the primordial sequence, which is completely ordered, 100% RNA repeats and inverted repeats. So this is a table of statistics that I'm not going to go through. So basically what it says is that every feature of the theorem as I've presented it to you is correct. I'll give you more details about the theorem shortly. Oh, there it is. So basically have an ancient polymer world. It's made up of polymers, which includes regular polymers. These are the ones that have survived in transfer RNA. And then you go to a 31 nucleotide mini helix world. So there can be mini helices of many different types, but these are the three that survived, and they survived in transfer RNAs. This is a type 1 transfer RNA, that's a type 2 transfer RNA. These are the primordial sequences. And there's absolutely no ambiguity of any of these sequences except in the red segments. Okay, so this sequence and this sequence, I don't know what the primordial sequences were, but I know what the sequences were at the time of the last universal common ancestor, which is also about 4 billion years ago. This is type 1, that's type 2. They vary only by the length of the variable loop. Here it's 5, here it's 14. 14 is 7 plus 7, 3' acceptor stem ligated to a 5' acceptor stem, like that. And here it's 5, it's those 5 after deletion of these bases, and there's the deletion there. So transfer RNA type 2 and type 1 come from processing of a 93 nucleotide precursor. But 93 is 3 times 31. So if you ligate this to that, you attach here and you attach there, you get this molecule. If you process here, here, and here, you get back more mini helices. So this is an intermediate in replication of mini helices in mini helix world. And if you make this 9 nucleotide deletion here, you get this sequence GGCGG, 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 GGCGG. That's a fragment of a 5' acceptor stem. And on this side, if you keep this sequence intact and you don't process it, you get a type 2 variable loop. And it's initially 14 nucleotides, as I sort of showed you, with 7 plus 7, 3' acceptor stem ligated to a 5' acceptor stem. And here it's been reduced to 5. It's those 5, CCGCC, 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 a fragment of a 3' acceptor stem is the type 1 variable loop. So 4 points to make here. The first point is what you see on this slide is 100% ordered sequence. And it's all RNA repeats and inverted repeats. The only slight exception would be this ACCA primitive adapter sequence, which gets attached to RNAs to attach amino acids. And then here, let's see. Second point, the anticodon 17 nucleotide stem loop stem and the T17 nucleotide stem loop stem, those are homologs. What I'm saying is that this stem loop stem and this stem loop stem, initially, those were identical sequences back here in mini helix world. And there's some ambiguity about the sequence of these loops. And these are specialized loops, as I'll try to describe in a minute. But anyway, these 17 nucleotides and those 17 nucleotides, after 4 billion years of separation in this molecule, they're still recognized and recognizable as homologous sequences. The same thing here, stem, loop, stem, homologous stem, loop, stem. So this sequence is genetically related to that sequence as indicated by the coloration scheme. Third point, you need 3 mini helices. So you can count them here, 1, 2, 3. You can count them just from cores, 1, 2, 3. These two are initially identical. This one becomes the anticodon loop of tRNA. This one becomes the T loop. This one becomes the D loop of tRNA, as indicated in the model. And fourth point, I've completely described evolution of type 2 and type 1 tRNAs, even though this whole process occurred 4 billion years ago on Earth. So you can use this as a cheat sheet to solve the puzzle. This is all chemical evolution, all right? So there doesn't have to be any divine intervention here. All you need to do is have a system that relies on polyglycine. This is a primitive method to synthesize polyglycine. This is a better method to synthesize polyglycine. And initially, this is the method to synthesize polyglycine, but this system can learn to encode proteins. So we've spoken about the God loop within the God molecule. So that's the anticodon loop. And this is really a highly specialized loop, and it was clearly chemically selected in the ancient world. It's a 7 nucleotide loop. It must be 7. Can't be 3, can't be 4, can't be 5, can't be 6, can't be 8, can't be anything but 7. Has to be 7. I'll try to make this point. It has a U-turn between loop positions 2 and 3. It's the most important biological intellectual property on Earth for evolution of life. So we have something resembling intellectual property before we have intellect. So basically, chemical evolution is driving this process forward. The genetic code evolved around the tRNA anticodon. And I'll try to make that point as well. So here we have our loop, our 7 nucleotide loop. This is part of the stem, that's part of the stem. The reason there are no hydrogen bonds here is this is a change to a pseudouridine, so it's been opened up to increase the dynamics of the loop. Here you have an A interacting with a methylated C by a Hoogsteen base pair. Here you have U33, and then you have follow that by the U-turn, a U-shaped turn in the RNA backbone, followed by a wobble base, 2 Watson-Crick bases. At a Watson-Crick base, you have 4 possibilities. So in terms of coding, this can be A, G, C, and U and recognized as such, and this can be A, G, C, and U and recognized as such. This position can't be A. It can be G, C, or U, and it's only recognized as purine versus pyrimidine. So there are really only 2 possibilities at a wobble base. So wobble is 2 possibilities, Watson-Crick 4 and 4, so 4 times 4 times 2. The maximum complexity of the genetic code originally evolves to be 32 assignments. But we only got 20 amino acids plus stops, and there are reasons for that as well, although that's a problem for another day. And then there's a heavily modified base here at 37. That's part of the reading of this base. So early in evolution, you have 2 wobble positions. And this one evolves partly through the modification of this space to be a Watson-Crick position. Also, there's an opening and closing mechanism of the ribosome that locks down this base and forces it to be a Watson-Crick base instead of a wobble base. Anyway, here's a codon, anti-codon table. So the codon, first position, second position, third position. And the anti-codon, that's the transfer RNA, anti-codon, 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 amino acid, and amino-acyl tRNA synthetase. So there are a whole bunch of points to make about this table, and I'll just make a few of them. First of all, there's lots of evolution around the 35 position. So what's the 35 position? That relates to the columns 1, 2, 3, and 4. This is the 34 position, that's 35, and that's 36. So at 34, pyrimidine versus purine, and never A. That's why the A is in red. And 35 is always C in column 4, always U in column 3, always G in column 2, and always A in column 1. And the 36 base, that relates to rows. So you have rows 1, 2, 3, and 4. But the code seems to fill by rows. Row 4, followed by row 2, followed by row 3, followed by row 1. So this is the favored row. That's the favored row because it's 36C. So it turns out that at this base and at that base, C is the preferred nucleotide. And so you fill row 4 first and then row 2. That's 36G, G, G, and G. And then U, row 3. And finally, row 1 is filled last of all. That's 36A. 36A is slow to fill because while this is a wobble position, you can't use row 1. You can't use 36A. So the wobble base is either G. G can interact with U as a wobble pair. G can interact with C. This is messenger RNA as a Watson-Crick pair. Or you can have U and C. U reads both A and G, and C reads G fairly specifically, but not specifically enough so you can encode 2 amino acids using U and C. So only 2 possibilities, purine and pyrimidine, at this position. So without this loop, you can't have a 3 nucleotide code. So why do I think that that's true? So this base here is heavily modified. It can't be part of the anticodon, so you can't have a 4 nucleotide register. Has to be 3. And this is a sort of tight loop. So it's selected chemically as a tight loop to present 3 nucleotides. And that's part of why this is such an important loop in evolving life on this planet. And I would say without this RNA loop, you would never get life as we know it on this planet. Let's see. So columns 1, 2, and 4 evolved by one mechanism, column 3 by another. Because column 3 evolves around the wobble position, you get separation in A and B rows. So you get aspartate, asparagine, and histidine, 3 related enzymes. And here you get 1B enzymes. Those are the charging enzymes that put the amino acid on these tRNAs. So that's what gives you the stripe pattern. But here you have larger sectors. You have leucine, which is a 6 codon sector, serine is a 6 codon sector, and arginine is a 6 codon sector. The large sectors are determined by fractionating along the 35 base, That gives you evolution in columns, and the 36 base, which is evolution in rows. So anyway, there's a great deal of order to the evolution of the genetic code. We'll discuss this in more detail later. Maybe that's too much detail for now. Anyway, I'm not sure. But anyway, I want to emphasize the importance of this loop for evolution of life on this planet. Without this loop, you would never succeed in evolving life. So according to the story I've told you, polymers go to mini helices. This involves regular polymers, CGC repeats, GCG repeats, UAGCC repeats. Those survived in tRNA, and stem loop stem, as you see in the anticodon loop and the T loop. And then you go to mini helices, and this includes 31 nucleotide mini helices, and these are highly ordered. And then you get primordial tRNA type 1 and type 2 with different length variable loops, but otherwise those are identical molecules. And these primordial tRNAs go forward into tRNAomes and the genetic code and amino acyl tRNA synthetases and proteomes, and these are all co-evolved. And once you co-evolve these systems sufficiently, you have a functional genetic code and you can have a living system with some other additions. So life on Earth and origin of life is largely a solved problem, at least at the outline stage. We have a really strong outline for how life evolved on this planet. And this story that I'm telling you will never be overthrown. So it can be criticized, but not in a logical way. It's obviously true because of the sequences of tRNA, because of the 331 nucleotide mini helix theorem. That's my Substack, so it's free and you're welcome to join. I explain how life evolved on Earth, and it's a good puzzle. And that brings us back to the top. And so, how are we doing, Lei?
Very good. I have a few questions I would like to ask, so can
Sure, shoot.
help the audience as well. So the first question, what do you think there are type 1 tRNA and type 2 tRNA?
Well, I think that having some type 2 tRNAs helped particularly with building an intact tRNAome to encode enough amino acids. So having some diversity in the organization of the variable loop allowed that to happen, to add more amino acids to the code. If they were all type 1 tRNAs, that might have been more of a problem.
So following that, why are there more type 2 tRNA? Why are most tRNA type 1?
Okay, so I sort of counted them up. So the top number, according to my understanding, is 3 for type 2 tRNAs. So in bacteria, it's tRNA tyrosine, tRNA leucine, tRNA serine. In archaea, which are an older organism than bacteria, it's tRNA leucine and tRNA serine. And so you really, and they all have to have a different score, and there are only 3 scores, 0, 1, and 2, which has to do with the trajectory of the variable loop arm coming off the body of the tRNA. So there are only 3 set points that are allowed, and each tRNA has to have a different set point. Otherwise, there's confusion about tRNA charging, among other potential problems.
So the maximal number of type 2 tRNA are 3, right?
That's right. So there can't be more type 2 tRNAs. And it turns out now there can't be any more type 1 tRNAs, really.
Right.
Except in special cases. Special cases, you can have other tRNAs.
So you say solve the tRNA using puzzle, like the puzzle solution. It does seem easier for type 2 tRNA. Why is that the case?
I think you have to repeat that question because somehow I got lost.
Okay, so the puzzle solution seems easier for type 2 tRNA. Why?
And why is that true? Is that
Right?
the question? Okay. So type 2 tRNA is simpler than type 1 tRNA because it only has one 9 nucleotide deletion. Type 1 tRNA has two 9 nucleotide deletions. Actually, those are related deletions, which is interesting. If they were on opposite strands of the RNA, they would both be identical deletions, actually. Um, but type 2 tRNAs with longer variable loops, the variable loop of 14 nucleotides is not processed.
See. So you mentioned polyglycine. Do you have any suggestion of experiments to do with using polyglycine?
Yeah, I think um, people could do a whole bunch of things with polyglycine. So I'm not positive that I know what's going to work best, but I would try to take polyglycine and see what kind of chemistry you can do with it using like an ancient environment, like a reducing environment. See if you can cross-link polyglycine chains together to make longer and more complicated arms. I think that um, polyglycine will probably help to make better protocells. So alongside the processes that we've described here, you have to be evolving protocells, which are not true cells, but are on their way to becoming true cells. I bet polyglycine would become a very important component of protocells. So I would try to do any kind of chemistry that you can possibly do with polyglycine and figure out how you could use that to build better and more complex systems in a pre-life world.
So my next question is there a God? Is a God necessary?
Yeah, so I don't know if there's a God. It's not really a very good scientific question, so I'm kind of agnostic about that. But a guide does not appear to be necessary to chemically evolve tRNA. Looking at the sequence of tRNA, it appears that it was chemically evolved, and it appears that we now know a whole lot about the process.
So how can pre-life primitive catalyst synthesize RNA repeats?
Um, right, so that's a good question. So, and I don't have a laboratory that's equipped to do that job. Um, but to make repeats, so the repeats that become tRNA could potentially be generated by a telomerase-like enzyme with a guide RNA to synthesize the repeat segments. You could have a system to spew out short RNAs that then could be ligated together to make repeats. That's another way to do it. You can have some kind of RNA slippage so that after you make one repeat, you slip and then make another repeat on a template. That might be similar to a telomerase reaction. So those are some ideas. I think somebody has to go into a laboratory and try and make repeats. I mean, it's absolutely clear from tRNA sequences. The tRNA was made 100% by repeats and inverted repeats. So that means that ancient catalysts made repeats. So it's just really a matter of figuring out how to build such a thing as a ribozyme or a more complicated primitive catalyst.
So what's the meaning of pre-life stem loop stem or inverted repeats?
The meaning, oh, okay. So, um, so if you have a stem loop stem, that's an inverted repeat, and that inverted repeats are used as replication primers to make complementary replicates. So if you have an RNA strand and you ligate a stem loop stem to it, you can, if you have a ribozyme or a primitive catalyst to make a complementary strand of the RNA, you can generate that. So one thing to recognize, so in tRNA, you have CGC repeats, you have GCG repeats. That's an indication that you have ability to make complementary sequences. And you have an ancient stem loop stem, which then later appears in the tRNA as a T loop or an anticodon loop. Those can be replication primers. Those also could be made by snapback replication by a complementary synthesis apparatus. So those are some ways that you could utilize stem loop stems.
One more question. Where were these models for tRNA evolution discovered earlier?
Yes, I don't know the answer to that question. So it turned out that this model was pretty easy to come up with compared to what I thought it would be. So I set out trying to look at tRNA evolution, thinking tRNA evolution was an impossible problem. But it turned out that it was very easy just using online tools to figure out that the T loop and the anticodon loop were obvious homologs. And if you solve that and you solve the acceptor stems or GCG repeat and the CGC repeat, you have most of the solution of tRNA there. Some of the other parts of the model were a little bit slower to work, but pretty straightforward. So it wasn't actually a very hard puzzle. And once we got started on it, it was pretty straightforward to solve.
Thank you, Zach.
So we're all done, or is it time to sign off?
Sure, maybe you can stop recording.
Okay. Yeah, I can try to do that. Oh, here.